Imagine you've just spent 2 hours studying something important, a language, a skill, material for an exam. You feel good about it, so you do what feels right. You go for a run. According to the research, that was a mistake. Not because exercise is bad for the brain, it isn't, but because timing matters in ways that most people have never been told. And that's just the first of 5 findings about intelligence and cognitive function that the research gets right and common sense gets wrong. Picture 3 groups of people. All of them learn the same set of picture location associations over 40 minutes. Then they split up. Group 1 exercises immediately. Group 2 exercises 4 hours later. Group 3 doesn't exercise at all. 48 hours later, they're all tested inside an MRI scanner. The group that exercised 4 hours after learning remembered significantly more than both other groups. And the brain imaging showed why. Their hippocampal activity during recall was more precise. The memories were encoded more cleanly. This study was published in Current Biology and has since been replicated. The finding is real. Here's the mechanism. When you exercise, your body releases epinephrine, adrenaline. Epinephrine plays a direct role in memory consolidation. It enhances synaptic plasticity and long-term potentiation in the hippocampus. The 4-hour window appears to align with a critical period in the early stabilization of new memories, the moment when they're most susceptible to being strengthened. Exercise immediately after learning doesn't give that window a chance to open. The adrenaline arrives before the consolidation process has begun properly. 4 hours later, it arrives exactly when the brain needs it most. There's a second layer to this. Emerging research suggests different types of exercise serve different cognitive purposes. High-intensity interval training, HIIT, appears to produce the most significant improvements in executive function. Your ability to switch between tasks, hold information in working memory, and suppress irrelevant information. Moderate intensity continuous training, a sustained jog, a steady bike ride, appears more effective for memory consolidation specifically. The research is still developing, but the direction of the evidence is consistent. The type of exercise and the timing of exercise both matter, and treating them as interchangeable misses something important. Most people understand sleep as a kind of off switch. The brain powers down, consolidates a little, and wakes up refreshed. That model is wrong, or at least severely incomplete. Here's what's actually happening. During slow wave sleep, the deep non-dreaming phase that dominates the first half of the night, your hippocampus replays the day's learning, not metaphorically. The same sequences of neural activity that fired when you learned something new are firing again during sleep, in compressed form, transferring that information from short-term hippocampal storage to long-term neocortical storage. Think of it like moving files from a temporary folder to a permanent drive. The hippocampus is the temporary folder. Sleep is when the transfer happens. A 2025 study confirmed that sleep actively counteracts the memory decay that occurs during wakefulness. It's not passive protection, it's active consolidation. The consequence of this is direct. If you learn something and sleep, you retain meaningfully more than if you learn something and stay awake. Learning in the evening immediately before a full night's sleep may be closer to optimal than learning in the morning and spending the next 16 hours awake. But here's what most people miss. It's not just the hours of sleep that matter, it's the architecture. Alcohol disrupts slow wave sleep, late eating disrupts it, inconsistent sleep timing disrupts it. 8 hours of disrupted sleep is not the same as 8 hours of intact sleep, and the difference shows up directly in what you retain. Sleep deprivation does something even more fundamental. EEG research shows that a specific pattern of neural activity beta desynchronization that marks the brain successfully encoding new information is disrupted after a poor night's sleep. The sleep-deprived brain doesn't just remember less, it learns less in the first place. Most people, if they think about omega-3 at all, think of it as a heart supplement, something in a capsule that their doctor vaguely recommended. The neuroscience tells a different story. DHA, docosahexaenoic acid, makes up more than 90% of the omega-3 in the brain, and it comprises 10 to 20% of the brain's total fat content. Let that land for a moment. A significant fraction of your brain's physical structure is built from a fat found primarily in oily fish. Your brain is not just influenced by DHA, it is substantially made of it. DHA is integrated into the membranes of every neuron. It determines how fluid those membranes are, which directly affects how efficiently electrical signals propagate between cells. It influences neurotransmitter synthesis, it reduces neuroinflammation, and it supports the growth of new neurons in the hippocampus. A separate analysis found that people with the lowest omega-3 concentrations in their red blood cells showed the worst outcomes on tests of brain structure and cognitive performance at midlife. And critically, the damage wasn't distributed evenly. It was concentrated in the people with the lowest levels. This isn't a story of marginal gains. It's a story of meaningful risk for people who are chronically deficient. Deficiency is common. DHA comes primarily from marine sources, fatty fish, seafood, algae. It doesn't come meaningfully from plant-based ALA sources like flax seeds, because the body converts very little ALA into DHA. If oily fish isn't a regular part of someone's diet, their brain's primary structural fat may be genuinely in short supply. For most of the 20th century, neuroscience operated on a simple and somewhat depressing assumption. The neurons you're born with are the neurons you die with. The brain was understood as a fixed structure. Connections could be strengthened, but the fundamental hardware couldn't be replaced or expanded. In 2025, a team at the Karolinska Institutet published research in Science that effectively closes the debate. Using single-cell RNA sequencing and flow cytometry, techniques that can identify cells by their molecular signatures with extraordinary precision, they found active neural progenitor cells and newly formed neurons in the hippocampus of humans ranging from 0 to 78 years old. 78 years old, growing new neurons. The hippocampus, as this video has made clear by now, is the structure at the center of learning and memory. The fact that it retains the capacity for neurogenesis throughout human life is one of the most significant revisions in neuroscience in decades. What drives it? Aerobic exercise is consistently the most powerful known stimulator of hippocampal neurogenesis in humans. Even a single bout increases BDNF, brain-derived neurotrophic factor, which is the primary molecular driver of new neuron growth. Slow-wave sleep creates the hormonal environment in which new neurons can mature and integrate into existing circuits. Genuine intellectual challenge. Learning something that actually requires effort reinforces and expands the circuits being used. What inhibits it is equally clear. Chronic stress, elevated cortisol, suppresses BDNF, reduces hippocampal volume over time, and directly impairs the growth of new neurons. There is a straight biological line from sustained psychological stress to structural changes in the brain's learning and memory center. The brain is not fixed. It never was. Each of the 4 findings so far stands alone, but the most striking evidence in recent cognitive research isn't about any single lever. It's about what happens when you pull all of them at once. The US Pointer trial, published in JAMA in 2025, enrolled around 2,000 adults in their 60s and 70s. All of them were sedentary at the start. Half were randomly assigned to an intensive 2-year lifestyle program, aerobic exercise, Mediterranean-style diet, blood pressure management and structured cognitive training. The other half were simply advised to eat better and move more. At the end of 2 years, the intensive group scored significantly better on standardized tests of thinking and memory. Their cognitive scores were equivalent to those of people a year or two younger. That is not a marginal difference. That is a measurable reversal of brain age through lifestyle alone. What makes this so compelling isn't any single component, it's the convergence. Exercise stimulates neurogenesis, sleep consolidates new learning, DHA provides the structural material for efficient neural signaling. Intellectual challenge drives plasticity, stress reduction removes the primary inhibitor of all of the above. These aren't 5 separate dials, they're one system, and the system responds. None of this is without nuance. The 4-hour exercise timing study used associative memory tasks, picture location pairs. Whether the same window applies to all types of learning, all levels of complexity, all ages is not yet confirmed. The finding is real, its generalization is still being mapped. The neurogenesis research in humans, while now significantly strengthened by the 2025 Karolinska study, remains harder to study in living humans than in animal models. We can see the precursor cells, we can see the immature neurons. What we can't yet fully measure is how functionally significant those new neurons are in real cognitive terms. The omega-3 findings are most relevant for people who are genuinely deficient, and the evidence doesn't strongly support additional benefit from supplementation in people already getting adequate DHA from their diet. And the Pointer trial required a level of support, structure, and intensity that most people won't replicate independently. The headline finding is real. The conditions that produced it were not ordinary. None of this changes the direction of the evidence, but it moderates how confidently any single finding should be applied to any single person. Based on the research, here's what the evidence points toward. Exercise timing matters more than most people realize. 4 hours after learning appears to be the window where aerobic exercise most effectively consolidates what you've just studied. For day-to-day cognitive function, HIIT and sustained moderate cardio serve different purposes, worth knowing when planning your week. Sleep architecture, not just duration, is where memory consolidation happens. The slow wave phase is when your hippocampus does its filing. Anything that disrupts that phase, alcohol close to bedtime, inconsistent sleep timing, late eating, reduces the cognitive return on your learning. DHA from dietary sources first. Oily fish 2 to 3 times a week covers most of what the research targets. For those who can't get that consistently, a marine-sourced omega-3 supplement covering at least 500 milligrams of DHA daily has a reasonable evidence base. Genuine novelty drives neuroplasticity. The brain responds to actual challenge, not familiarity dressed up as learning. Passive consumption of familiar content doesn't produce the same neuroplastic response as genuinely difficult material. And chronic stress is the single most direct inhibitor of the neural growth mechanisms this video has covered. Cortisol suppresses BDNF, the primary driver of neurogenesis. That's not a wellness platitude, that's a mechanism. To summarize, the research on cognitive function points consistently toward the same levers: exercise with smart timing, sleep architecture intact, adequate DHA, genuine intellectual challenge, and chronic stress kept in check. None of these are new ideas. What is new is how precisely we now understand the mechanisms, and that precision changes how you think about them. The brain that existed in neuroscience textbooks 30 years ago, fixed, slowly declining, largely beyond influence, doesn't match the organ the evidence is describing now. It responds, it adapts, it builds new tissue at 78. If you found this useful, subscribe. I cover research like this regularly. breaking down what the actual studies say rather than what the headlines want you to believe. I'll see you in the next one.